In this video, we will discuss about whether DiaBZl will promote tumor apoptosis or not. In the previous video, we have understand the role of DiaBZls on the engineered T cells. That is, it will cause enhancement of the STING signaling pathways. At the same time, it will also increase T cell receptors signalings. That helps in the identification of the tumor cells. In this video, we will discuss the effect of DiaBZls on the tumor cells. So what they have found out is that DiaBZl will cause or will promote tumor apoptosis, and how they come to this conclusion we will understand through this figure, results that are presented in this figure. So first, what they have seen is this cytotoxicity effect of the DiaBZl on this MEL 526 cells, and what they have found out is that after 6 hours they can see their cytotoxicity is increases, but after 3 hours they do not see there is an increase in the cell cytotoxicity. But for the 6 hours 24 hours they can see that increase in the cell cytotoxicity compared to the control case where DiaBZl is not, whereas MEL 526 cells are not treated with DiaBZls. When they check the different apoptotic factors, what they have found out is that phosphorylated TBK1 and phosphorylated H2AX is increased with increase in time. However, there is no change in these NY-ESO expressions. I will come to this point in the end of this video, but for the now let us just understand that NY-ESO does not change with the DiaBZl treatment at different time intervals. When they check the quantifications of the Western blots that are shown in the panel B, they can see that for this phosphorylated PTKB is higher at 3 hours, but it will go down at the 6 hours. But for the case of this PH2K, they can see that there is increase in this PH2KX with increase in the time of this DiaBZl treatment. And once validating this results that DiaBZls will cause activations of this signaling pathways, that is phosphorylated H2KX and phosphorylated TBK1, but phosphorylated TBK1 will go down after the 6 hours, but it will goes up at 3 hours. And thereafter they check the different pro-apoptotic factors, and what they find out is that apoptotic factors such as caspase-3 and CL caspase-3 increases with increase in the increases in the case where DiaBZl treatments occurs. For example, this cleaved PARP is higher with increase in time. However, for the case of this cleaved caspase-3, it is higher for the 3 hours, but it will go down for the 6 hours. But the difference between 3 hours and 6 hours is not statistically significant, but both of them are higher than the control case. And after that they wanted to check this interleukin 6, which will cause this the pro-apoptotic factors. We can see that for the case of this 1 microgram DiaBZl 10 microgram DiaBZl treatment, they can see that it is significantly higher compared to the fold change is significantly higher compared to the control. But for this H1F1, they do not see an increase in the this interleukin 6. Interleukin 6 are the pro-apoptotic factors that will attract immune response, and other we can say pro-apoptotic factors such as PUMA and VAD that will compensate the anti-apoptotic factors is also shown to be increased at 6 hours, whereas this increase is not significant at 3 hours, indicating that with increase in time there is a pro-apoptotic factor increases. That is shown here for this PUMA, VAD, and also for the NOx. For the NOx, we can see that with increase in the DiaBZl concentrations, there is an increase in the NOx formations. However, for the H1N1, they do not see any increase in the NOx formations. Now this you can consider it as a control. So all this indicates that this DiaBZl has a specific role towards promoting this tumor apoptosis. And however, there is a expression of this NY-ESO on the tumor cells does not seems to be changed for the DiaBZl. However, we will see in the subsequent videos that when they measure NY-ESO through immunofluorescence, they can see that the NY-ESO changes with change in the DiaBZl treatment with respect to time. So one reasons that they have mentioned in the paper is that the antibodies is not capable enough to detect the increase in this NY-ESO, and that changes will reflected in the immunofluorescence assay and the other complementary assay that we will discuss in the subsequent videos. That's all for this video. I hope you understood how they made a point that DiaBZl will promote tumor apoptosis. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.